This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 294, recorded on August 17, 2023. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Charleston, South Carolina, Michael Schmidt.
Well, hello, everyone.
Where you have a hurricane, right?
Not yet. Everybody is gone and lit a candle at the cathedral, hoping it stays out at sea.
Why are you still there, Michael?
Well, it hasn't been announced yet. There are two storms out off the Horn of Africa that the weathermen are having conniptions over.
Michael is devoted to TWiM. He would stay there until the end.
When are they anticipated to make landfall?
They don't think they will. They think that they're gonna.
Okay.
They think they're gonna peter out. But
Great.
Since it's the first ones of the season, they get awful excited. And we had our hurricane preparedness drill at lunch today, so everybody knows how to close down their labs and how to protect their animals and all those things.
I'm sorry. I'll be thinking of you all.
Thank you.
Also joining us from Ann Arbor, Michigan, Michele Swanson.
Hello, hello. It's not hurricaning here. We're safe.
Yeah, it doesn't do that there, right?
Nope.
You're too far north. You get snowstorms.
But we have the Great Lakes buffering us from the worst of the heat fluctuation, too. So I'm pretty happy to be here.
Before we get into our papers, two things I want to tell you. We depend on your support for doing these programs. If you enjoy it and you'd like to support us, go to microbe.tv/contribute. And in addition, we've added a new podcast to the Microbe TV stable of science programs. It's called Matters Microbial.
Yes.
It is a product of Mark Martin, and Microbe TV is producing it for him. So you should go check it out, microbe.tv/mm. By the time you hear this episode of TWiM, there'll be maybe 4 or 5 episodes. He's putting one out a week. He's had me as the first guest. He had Myra Breitbart and Seth Bordenstein. That's right. Seth Bordenstein, his third guest. So check it out.
Yeah, in general, the format is he wants to identify thought leaders in the field and have them come on and talk about their research. So
yeah, and 20 minutes or less. That's the idea. I said keep it 20 minutes or less, and that'll be good. So check it out, microbe.tv/mm. All right, it's time for some ice cream, Michael.
All right, so the title of today's snippet is Psychrotrophic Bacteria Equipped with Virulence and Colonization Traits Populate the Ice Cream Manufacturing Environment. It's from Applied Environmental Microbiology, and it was published in August. It's from a group in Naples, Italy, by the first author is Vincenzo Valentino. The second is Francesca De Filippis. And then we have Giuseppina Sequino and Danilo Ercolini. And they are from I probably butchered their names and I apologize. They are from the Department of Agricultural Sciences at, as I said, the University of Naples. First rule, when hunting for appropriate papers for TWiM, try not to be craving that delightful summertime treat of ice cream. Now, seriously, the topic of today's paper dovetails well with my own research interests, namely, how do we protect ourselves from our microbial friends when they and we are interacting with the same things, namely the built environment, whether it be in a clinic, like I focus on, or food preparation services from the deli slicer that delivered that delicious sub I had for lunch, or to that evil soft serve ice cream maker that I find myself always gravitating towards if I'm good and don't partake of too many samples during my weekly adventure to Costco. In fact, many microbial species can permanently establish themselves within and on food processing surfaces, thus contributing not only to food loss, but actually a risk to the individuals who actually consume the adulterated food. Additionally, not only do the contaminated microbes add to spoilage, but they also can be transferred to intermediates, other surfaces, other products that we ultimately consume, thereby representing a potential human health hazard. So today's snippet will introduce us to the issue of the built environment, albeit not in a hospital setting, but rather in things that we all interact with. And one of the areas is one that we might least expect, namely the devices responsible for creating that delightful summertime treat of ice cream. Here, the authors will provide evidence that even in spite of employing very prescriptive and often very rigorous sanitation schemes, complex microbial communities can and do exist in places where we may never suspect them taking up residence. And in fact, today we're going to learn all about the microbial communities affiliated with an ice cream producing facility in Italy.
And can I just say, if you're a big ice cream fan, you might want to go find something else to listen to.
Oh, Michele.
No, this is troubling.
But people don't get that. Only immunocompromised people should worry, right?
Only, yeah.
Michael, what does psychrotrophic mean?
It means they love to grow in the cold. So the microbes that are psychrophiles will actually expand and actually add to the volume of microbes present in the material. So they're going to continue to grow. And let's face it, ice cream is a wonderful food source.
What would be the doubling time, Michael, at
it's slow.
4
it's
degrees?
Slow. It's
like once
slow.
A day?
No, it's on the order. It's still in minutes, but it's a long time, generally on the order of in about 60 to 90 minutes doubling time. But, some of these microbes that can grow psychrophilically, like Listeria, are especially problematic because their infectious dose is so low. You only need to get a few of them in order to cause problems. And if you happen to be immune suppressed, it can cause a great deal of trouble for you. So I recently was talking to one of our longtime listeners of TWiV and TWiM, and he challenged me. He says, TWiM is sometimes hard to listen to because sometimes you guys forget to explain what the abbreviations mean and what some of the techniques do. So in keeping in what Sewell offered, he said, today's paper uses a lot of abbreviations. So I'm just going to give you a cheat sheet. FC corresponds to food contact surfaces. NFC is a non-food contact surface. A MAG is a metagenome assembled genome, which is a single taxon. And a taxon could mean a species, a genus, a family, or even a class of microbes that was assembled based on one or more binned metagenomes, where the process of binning collects or groups sequencing reads that come off of these fancy instruments that many of us now have in our labs that are often called in the jargon contigs, where then the binning software assigns them to an individual genome. The final abbreviation I'm going to introduce you to is one that is critical to really appreciating the data that come out of this paper, and that's TWiM. TWiM is nothing more than a user-friendly pipeline for whole bacterial genome analysis from any bacterium from any origin generated as a consequence of these new high-throughput sequencing apparatus like the one from Illumina. TWiM was developed for the Illumina sequencing platform, and recall that Illumina determines the sequence of nucleic acids through a process that they term sequencing by synthesis. So once completed, the TWiM software generates, and this is the key, an interactive report that is platform agnostic, which means it will open on any web browser so you can share your work with others while allowing you to ask questions like the authors did here. One final piece that Michele sort of alluded to is, you may think talking about ice cream is a bit silly, but back in 2020, the United States Justice Department fined Blue Barrel Creameries $17.25 million in criminal penalties in connection with listeria contamination from 2015. That's a
from multiple states. Multiple states.
From multiple
too.
states.
Yeah.
And people died from it, and it was really quite tragic. And what this should reassure consumers of ice cream across the globe is that not only here in the United States, but in Europe as well and other countries, such a criminal sentence sends a clear message to food manufacturers, at least here in the United States, that the Department of Justice is taking appropriate actions when contaminated food products endanger consumers. So this really should give us some comfort that when we eat that ice cream, we can rest assured it's going to be safe to eat. Now, the methods used to answer the ever-present question, or the CSI, if you will, of what's going on in this ice cream plant, is they recovered the total nucleic acid from swabs. They went out into the ice cream plant and swabbed not only the surfaces that was manufacturing the ice cream, but they also looked at the raw ingredients going into it. And they asked the question, who's there and how many are there, by processing the nucleic acid. And the screen that they used is they looked for the 16S ribosomal RNA gene. And as many of us can recall from our discussions of the microbiome, it's effectively the molecular signature that we in the microbial world use to tell the microbes apart when we're using simply nucleic acid. Now, this process, they yield over 1,000 taxa from 56 samples that they evaluated from an ice cream plant in Italy that they only investigated on a single day from July of 2021. They point out that the average temperature of this significant industrial plant was kept at 4 degrees. So now, please don't read the methods unless you have some chocolate-covered almonds or peanut M&Ms in your desk, because you're going to be scrambling like I did looking for them. So here the authors look at 2 manufacturing lines from their plant. They labeled them C and M, and they also looked at the raw ingredients that made up the 2 products. Line M produces an ice cream made of a mixture of cream and milk chocolate covered almonds. And the product of, I told you, the product of the C line is a thin wafer whose inner part is covered with a thin layer of chocolate and then filled with the ice cream. On top of that wafer, they put chopped hazelnuts. So that just set me over the edge.
Wow.
So rather than calling these lines M and C, I'm going to call the M line the almond line and line C the wafer line. So the first thing they did is they asked the question, who was there? And they recovered Pseudomonas. It showed the highest average relative abundance at about 7.5%, followed by another bad actor, Acinetobacter, and then, of course, Streptococcus.
Michael, the numbers, the percentage you're giving, what does that mean? 7% of what?
Well, that's the beauty of the Illumina sequencing process, because it's sequencing by synthesis. When you have the total nucleic acid that you recover, you have these adapters that get glued on. And into the show notes, I dropped a YouTube video from Illumina explaining how the sequencing by synthesis process works. So they glue these adapters on to the sheer nucleic acid, and within the adapter is a primer for the 16S ribosomal RNA gene that then gets amplified up. And so the proportion is based on the proportion of total bacterial nucleic acid that was amplified. So Pseudomonas was about 7.5% of the total bacterial DNA amplified. Remember, they had over 1,000 taxa or genus and species in this particular case. That was followed by Acinetobacter. Streptococci was between 4 and 13%. And the percentages differ because of the materials that they were sampling, whether it's the chocolate-covered almonds or the thin wafer material that they were using, or simply the cream itself, or, in this case, the surfaces. They also found...
Just to be clear,
Michele.
This is just at the genus level, right? So we don't know which of the Pseudomonas or which of the Streptococci.
Not at this point in time.
Right.
They delve into it more as they go further into TWiMs.
I don't want to freak people out and think that they're contaminated with human pathogens. Okay.
No. And they found one other interesting bug that I wasn't familiar with, and that's curvy bacteria. And here... Now, this genus is only 20 years old, and it's named pretty well because it's a curved bacterium. And this is a gram-negative heterotroph that happens to be microaerophilic to aerobic, and it's often isolated from well and distilled water. So this is an organism that's probably growing on the margins and just is affiliated with the surface. It's living on the fringe. And originally it's in the same family as the beta proteobacter, and it's moving up the taxonomic scale. You have the Commononis, so that's the family, and then moving up to the order, you have Burkholdereales. And so it's just like the Pseudomonas. So these are environmental critters that they are discovering. Now, the differences that they saw were most appreciated between the two production lines, if you will, the almond versus the wafer, as well as between the food contact surfaces and the non-food contact surfaces, which generated a very colorful figure 1, illustrating not only who was present, but again, they not only know who, but their proportion because of the method that they use to determine the who, because the sequencing by synthesis process allowed them to figure total nucleic acid of the microbes that were being released. Now, the food contact surfaces from the wafer line were highly populated with Pseudomonas and curvy bacteria, while the non-food surface lines showed a higher abundance of Rothia and Acinetobacter. Again, Rothia, if you're not familiar, is a gram-positive coccobacillus that has a wide range of infection opportunities, especially in immunocompromised hosts. And they are considered part of normal human flora of the oropharynx and upper respiratory tract. And they are, again, aerobic, facultatively anaerobic, non-motile, non-spore-forming gram-positive bacteria that can filamentously branch. The reason I emphasize filamentous is because these suckers are probably holding on to the surfaces, which is why we're beginning to recover them from our swabbing of the surfaces in this particular built environment.
Probably forming biofilms, and so they're probably very difficult to get off during the routine cleaning.
And remember, a lot of these lines are using the equivalent of pressure washers that are just like the pressure washer that you use to clean your deck, that actually add a little bit of bleach to it to try to liberate it. But these suckers hold on.
Yeah, so now, it's not sloppiness on their part.
No. It's not sloppiness.
Tenacious microbes that have evolved.
And a lot of these surfaces are food grade stainless steel. And while stainless steel looks shiny, it's actually, even if it looks shiny, it could be scratched. And those little scratches are where the biofilms attach themselves and hold on. Now, the almond line showed a high abundance, again, of curvy bacteria and Pseudomonas, but they also recovered some bacilli. And bacillus is significant because they form spores, which means they can hang around, and they're also refractory to many of the disinfectants that are used to clean things up. Another technique that this group used is principal component analysis, which is a popular technique employed by computational biologists simply for analyzing large data sets containing a high number of dimensions or features per observation. And you think about it, the surfaces that they looked at, the microbes that they recovered, we have a lot of data to compare. And what you will see in this second figure by their principal component analysis, and they were using a form of PCA called Bray-Curtis, or distance or dissimilarity. And they're asking, are the surfaces distinctly dissimilar? But unfortunately, they weren't able to discern surface differences according to the processing line that they were evaluated. That didn't surprise me, given my experience in the built environment, that even the most rigorous cleaning protocol, it's pretty hard to strip away microbes to really change things up. Further, they also subjected their data to Shannon and Simpson analysis, and Shannon diversity is a way to measure the diversity of species in a community, counting up how many are there. And Simpson is an index that measures the number of species present. So again, they're lucky that they use the Illumina process rather than just doing, full-scale shotgun of whatever was there, because they wouldn't be able to get a metric of how many. In spite of all this consideration, the Shannon and Simpson's diversity indices were not significantly different between the food contact regions and the non-food contact surface lines. And again, as Michele points out, it's probably because of the rigorous cleaning that they're doing. And once these suckers attach, they attach. And, the issue is keeping them at a very low level. Now, one last bit of analysis from the census of the two lines revealed that on average, and here's the trick, the ingredients contributed most to define the microbiota of the products associated with both lines, followed by the clean food contact surfaces. So again, you need to be certain about your raw ingredients that are coming into your plant. And so you have to do a QC to make certain that you're not getting contaminated, chocolate covered almond clusters that happen to be coming in. They also commented that the food contact surfaces were highlighted as the primary source of Pseudomonas and Curbivacter found in the final products of both lines, whereas the non-food contact surfaces from the almond line, it was most likely a consequence where it was represented significantly by Acinetobacter. Now, this next focus of their work is what I found to be most fascinating and addressed a new technique that I had been familiar with, namely TWiM analysis. Here's where this comes into play. They learned from their screening of the sequences, and they can go back and probe the DNA further, that the screen recovered 386 genes across 282 contigs. And remember, contigs is the jargon for the genus or the species that is associated with the organism that the virulence gene is associated with. In addition, 111 out of those 282 contigs were in high quality MAGs, or that metagenome associated genomes, with 23 of them being included in two MAGs that were attributed to one species of Pseudomonas stutzeri, and that were reconstructed from two surfaces from the wafer line. And here's where it gets really interesting. Now, both MAGs of the stutzeri showed genes were linked to biofilm formation, as well as genes related to pill synthesis and regulation. So that's code for not only are they making the genes to allow that initial attachment to the surfaces that are rigorously cleaned, but they also are encouraging the formation of biofilms. The other thing that they unfortunately learned was the ice cream factory community of microbes harbored 380 genes that represented antibiotic resistance traits, or the resistome, if you will, of the ice cream factory. It came as no surprise to me that the genes that were most often recovered that were antibiotic resistance traits were against beta-lactamase. We've used beta-lactam antibiotics that's things like penicillin, for well over 80 years. They were also then followed by fluoroquinolones, aminoglycosides, and cephalosporins. But the scary thing out of the TWiM data was that the antibiotic resistance traits were not only found in the chromosomes of the microbes, but they were also affiliated with plasmids, which is code for genetic exchange if those organisms get into things like our gut, or even exchange antibiotic resistance traits or other virulence traits within the community. So summing up, even at 4 degrees, we need to keep our food preparation areas pristine. Here, even at 4 degrees, bacteria were quite prevalent in spite of them cleaning rigorously. And they also, harbored some microbes that had the potential to be pathogens, and they harbored antibiotic resistance strains, with some of them being on plasmids. So a cautionary tale to all food producers that perhaps they need to understand the microbial community and their production plants. And I think this paper really goes to show the food preparation industry, they should do routine surveillance to figure out who's in their factory, to understand whether or not things are shifting towards a more pathogenic community or something that they can manage. They conclude offering that this was a one-off study, and even that causality is extremely hard to prove, and it's extremely expensive if you have to carry this out. But they're going to continue to rigorously clean, and I encourage them to change up the disinfectants. So just like we saw with bacteria becoming resistant to alcohol hand gels, we don't want them to becoming resistant to the cleaning solvents that they're using to disinfect our ice cream factories. So that's today's snippet.
Michael, let's talk about copper instead of stainless steel. Is it practical to convert to all copper material?
Not for food, because it typically reacts. And remember, even if you have copper cookware in your home, it's often coated with tin, because they don't want the copper reacting with the food, because many of our foods have acid in them, and that would liberate copper metal into our diet. And there's a fraction of the population that has a copper sensitivity. And so it would cause more problems than it would solve. I think hygiene is the key.
And tell them why I brought up copper. Why?
Copper is continuously antimicrobial, and it does have an application in the clinical environment. And we and others have demonstrated the utility of incorporating copper into common touch surfaces in a clinical environment. And it actually can reduce the incidence of healthcare-associated infections, at least in the study, the clinical trial we did, by 60% approximately.
So Michael, as you said, this is just a one-off, but let's say other studies are done and it turns out that this is a widespread issue. Then you are faced with the question, what do you do about it? Because if there aren't that many outbreaks, ice cream based outbreaks, the manufacturers are not going to want to do anything. And then it's up to the regulatory agencies to say, you have to do this cleaning and monitor it and so forth. And I could see
monitor
that
product.
They routinely monitor product, and that's effectively what got Bluebell Creameries into criminal trouble. Their leadership knew of the contamination of listeria in the product they were shipping to consumers, and in an email chain, the CEO said, "Ship it out; it'll be a limited problem."
Impact.
And that was a mistake. Yeah. But I mean, you can say, check your product, but then if you find contamination, you have to get it at the source. So
yes,
you have to,
I mean, this has happened. this has happened in the United States a number of times, not only for ice cream, but peanut butter. There was an incident a few years ago where a peanut processing facility in Georgia was contaminated by salmonella, and it's again over cleaning. And as you can imagine, peanut butter is ground at room temperature to keep the oils moving, and the salmonella just grew, and it was an odd salmonella. And there were, again, a number of illnesses, and they did a massive recall of peanut butter.
So, Michael, currently in the U.S., are there regulations as to what bacteria can be in ice cream and at what level?
Yes, the FDA has all sorts of guidelines that I'm sure we'll hear from our friends in food science all about.
So essentially, don't be scared to eat ice cream because it's highly regulated, right?
Well, especially since you're going to the land of Ben and Jerry's next week. I'm hoping you have Ben and Jerry's send us a couple of gallons.
Sure, no problem. I'll check it first.
Chunky Monkey or Cherry Garcia, any of those would be great.
All right.
I have to add, this isn't a problem just in the food industry, but water. Our municipal water supplies. I've studied Legionella for a number of years. It's naturally in our water systems, can form biofilms. So if the water isn't treated properly, or if it becomes stagnant because the water hasn't been used, then a bolus of Legionella can be spread through equipment that forms aerosols, and then most people will be fine, but those that are vulnerable can get ill with a pneumonia. So. Yeah. But if all the buildings test for it, then they're liable.
And
we don't spread it. We don't spread Legionella from person to person. It's always from a water source that should be clean. So it's a struggle.
Here in hospitals, we keep our hot water hot to effectively prevent the growth of Legionella in our hot water supplies.
Right. And you monitor the chlorine or whatever.
We monitor the chlorine. We monitor everything. Hospitals are very concerned about that, as Michele knows better than most.
We appreciate it.
Thank you, Michael.
Thank you.
All right, Michele, what do you have for us?
I have a paper entitled Panabacillus Species Infection Among Infants with Post-Infection Hydrocephalus in Uganda, an observational case control study. That's a mouthful, and after I introduce the authors, I will go into some detail there. But this was just published in Lancet Microbe in June of 2023. And the first author is Sarah Morton. She's a physician scientist who earned her MD PhD at UCSF and is now at Boston Children's Hospital. And then there are 4 senior authors, including the late Julius Kiwanuku from Lancaster University in the UK, James Broach from Penn State College of Medicine, Joseph Paulson, who's a biostatistician with NPower Medicine, a Bay Area startup that collaborates with physician scientists to conduct clinical trials, and then Stephen Schiff, a physician scientist and also the vice chair for global health at Yale School of Medicine. And among the 39 authors on this paper, who hail from 5 countries, are physicians from the Department of Pediatrics and Child Health at the Mombali Regional Referral Hospital, and also the Cure Children's Hospital in Mombali, Uganda, and also the Mulago National Referral Hospital in Kampala, Uganda, and scientists from the Department of Microbiology in Umbara, University of Science and Technology in Uganda. So this was a huge effort, and you'll see why. And I want to emphasize the authors are very grateful for the funding that made this research possible. In addition to getting some funding from Boston Children's Hospital Office of Faculty Development, they got a number of NIH Directors Awards, a Pioneer Award, and a Transformative Award. So these are special grants that recognize exceptional performance and special efforts beyond regular job assignments by funding high-risk and high-reward projects. And you'll see that this is a very large effort that required not only really thoughtful design at the outset, but also partnering with physician scientists from all over the world, especially in Uganda, to collect the samples that they needed to study this terrible infection. So the motivation is they were seeing this post-infection hydrocephalus in Uganda. So this, as the name implies, is an accumulation of cerebrospinal fluid in the ventricles of the brain. and it's preceded by a serious infection, typically neonatal sepsis. And in Uganda, there are typically 4,000 cases per year in infants. The only available treatment is neurosurgery to relieve the pressure that builds up from this fluid accumulating in the brain. And because that procedure is really limited in the resource-limited region of Uganda and Africa, this is often fatal in these neonates. So they were motivated, obviously, to figure out what's causing this. And what we'll learn is that they've identified a panabacillus. So this is a genus of rod-shaped facultative anaerobe gram-positive spore-forming bacteria, originally classified as bacillus, and that's why the term means almost bacillus. It's very difficult to culture by standard techniques, and what they found here, it's resistant to standard antibiotics, including the penicillins, but also vancomycin. So the genus, I've tried to learn a little bit about it. I've never heard of panabacillus. It's been isolated from a wide range of environments, including, I did a quick PubMed search, and there was a 2023 astrobiology paper that found it in samples from Mars. This is the genus now. Also honeybees, according to a microbiology research announcement, environments and habitats in various plants and worms, and occasionally causes human infections. And the risky ones are neonatal meningitis, but also wound infections. So what we'll learn here, that of 400 cases of this post-infectious hydrocephalus, that is fluid accumulating in the brain in infants in Uganda that they studied, 44% had this panabacillus in their cerebrospinal fluid as judged by PCR. And again, I want to emphasize that this particular syndrome is typically the patient will have neonatal sepsis, and then 1 to 3 months later will present with other symptoms that are indicating they've got brain infection and accumulation of cerebrospinal fluid in their brain can cause death. So to discover a candidate for this dreadful disease that's killing neonates in Uganda and other African countries, they did, as they said, this observational case control study. What that means is they recruited 3 different cohorts of people. They recruited 99 pregnant people, half who had fever during their delivery and half who didn't. Once they recruited them, they collected vaginal swabs and also blood samples from these 99 pregnant people. They also, in a separate cohort, recruited 800 neonates and were able to collect 750 blood samples and 630 cerebral spinal fluid samples that they will analyze. And then their third cohort was 200 infants who had this fluid accumulation in the brain, hydrocephalus, and 200 infants, neonates that did not. And from them, they collected cerebral spinal fluid, and we'll use that to do 16S RNA sequencing, which, as Michael pointed out in the last paper, is a standard technique to do genus level investigation of what microbes are present, if any. So in figure 2, they lay out in a very dramatic figure, panel A, who is there in the cohort of neonates that had this post-infectious hydrocephalus, and that's in the left panel, and they compared those to the control patients, neonates who did not. And what we see is there's this huge increase in the panabacillus identification by 16S RNA DNA sequencing in the cohort that has this syndrome. So they've got a candidate now. They then went on and used quantitative PCR of the cerebral spinal fluid to verify. So to go beyond just doing the whole genome sequencing or pan-genome sequencing. They went back and verified that, yes, indeed, there is 16S RNA and another gene typical of this panabacillus species. So next, they show in their table prevalence of this particular microbe in the blood samples of the patients that had sepsis versus the patients that had post-infection hydrocephalus. And they look also in the maternal blood. They didn't didn't have any positives there. They looked in the vaginal swabs of the pregnant people before delivery. Again, no evidence of this microbe there. They took specimens of the placenta after the live birth. Again, no indication, no detection of the microbe there. They also looked in either the umbilical cord blood collected during delivery and neonatal blood, and there was less than 1% of the samples from patients who had neonatal sepsis were positive for this particular genus. But for the cerebral spinal fluid samples, they got 5% of the patients with neonatal sepsis were positive for this Panabacillus, and 4% had the particular species that we'll talk about, thiamine liticus. But if they now compared that to the infants that developed months later, 1 to 3 months later, this swelling and accumulation of fluid in the brain, now 44% of the samples were positive for Panabacillus, and 41% were positive for not only Panabacillus, but this particular species, Thiaminolyticus. So they have a candidate now based on it being the most, the outlier, the distinguisher between the infants that went on to develop this serious disease and those that didn't. And the fact that they could not detect it in the maternal samples lead them to think that it's probably an environmental source rather than passage vertically from the pregnant person to the infant during delivery or during gestation. Any questions there so far?
Well, the one question I had is how is the umbilicus cared for subsequent delivery? Are they packing the umbilicus with any material that could introduce spores of this bacterium because some cultures actually pack the umbilicus with soil to effectively soothe the infant. I recall reading that when I was teaching tetanus, and some cultures actually pack the umbilicus with material. So that would be my one thought of how the Panabacillus could be introduced, because it is indeed a spore former.
Right. That's very interesting. And this group, as we'll see, and I'll emphasize at the end, is confident now that they've got a candidate that is associated with this dreadful illness. And their next step is to use the information they've learned and now to identify the source. And I think that's an excellent idea. If there's contamination of the water that might be used, or as you said, the spores during the delivery. So they want to investigate cultural practices around childbirth and neonate in these various cohorts that they recruited from. Good point, Michael. So they did want to now investigate the geographic distribution because they didn't have evidence that it was being vertically transmitted from the mother. So they looked at and plotted the distribution of these cases geographically. So are there particular regions of Uganda that have more cases than others? And that's shown in Figure 4. And again, they compare hydrocephalus that is not post-infectious versus that is, and those that are positive for this Panabacillus genus, and those are negative. And in general, they did see more clustering of this Panabacillus genus. And not only that, but they found it in regions of Uganda that are wet. So, for example, they found that cases tended to cluster in swampy regions on the northern banks of Lake Victoria, which is the source of the Nile River, and then also on the banks of another lake where the Nile flows in and out. And it also correlated, interestingly, temporally with the rainy season, more cases during the rainy season. So they are thinking that it could be an environmental source.
Could be something as simple as washing the baby the first time.
Could be. So they next did follow a few of the patients in detail to really probe we've associated this particular bacterium with these dreadful infections, but do we have evidence that they're the cause? So they include clinical follow-up on a few of the patients, and I'll just describe one. This was an 8-day-old baby who had came in with family, brought them in because the baby had fever, was irritable, was having seizures, and bulging in part of its head, which is an indication that they might have had swelling in the brain. They looked at the records, and the mother did report fever during labor. And so they then went on and did ultrasound of this infant. And those pictures are pretty dramatic, and they're shown in Figure 5, where they compare a healthy cranium to that of this 8-day-old patient who was reporting this irritability, and the mother had fever. And what they show in the figure are there are these regions that are clearly have abscesses and consistent with fluid accumulation. So they also did qPCR on samples that they collected both at 8 days and then 44 days later. So this infant was given antibiotic to treat the infection, but also was followed. And they found that indeed the samples were positive for this Panabacillus thiaminolyticus in both cases. So that is their evidence that this is a contributor to this dreadful infection that is unfortunately prevalent in regions of Uganda and that requires antibiotic treatment that is different from your standard antibiotics, the ampicillin, for example. But even that, the antibiotic treatment is not sufficient to relieve the pressure that builds up from this swelling, and you need to have neurosurgery done, which again is not readily available in these under-resourced countries. So the authors close by saying that identifying the source and routes of these infections is crucial for preventing neonatal sepsis and the consequences of post-infectious hydrocephalus due to this Panabacillus thiaminolyticus bacterium. And I will say, members of this cohort, this team, did publish in MBio in 2022 whole genome sequence analysis of 3 of the clinical isolates that were obtained during this study. And what they found is that the clinical isolates were distinct from environmental isolates in that they had a mobile genetic element that encoded a type 4 pili genes. And as Michael talked about in his presentation, pili are known to mediate adherence of microbes to surfaces. And in a number of pathogens, they're known to be a critical virulence factor. And in fact, in this MBio paper, they were able to show in a mouse model of infection that the strains that had the type 4 pili were more virulent than strains that were engineered to lack the type 4 pili. So there is hope. They have learned how to culture this. They actually use blood culture bottles, but they put cerebrospinal fluid in instead, and they are able to culture the organism. And that is, of course, going to open up the opportunity to do more molecular studies so that ideally they can develop a quick diagnostic and begin to learn where this organism is residing and how people are becoming exposed, and then who's infected and what antibiotics will need to be administered right away.
I'd like to point out that this is one of the most beautiful colony-forming type organisms that I have ever seen. And we'll drop into the show notes a colony generated by a chiral morphotype, a different species rather than the thiaminolyticus. This happens to be Dendritic and it's absolutely spectacular. It looks very science fiction-like, but this is a Panabacillus on a Petri plate.
It's beautiful.
It's quite impressive.
So this paper was just published in August, but a few weeks ago I received an email from the senior author, Stephen Schiff, who wrote an email to both TWiM and Daniel Griffin. He wrote that we recently cracked the mystery of what causes several thousand cases of post-infectious infant hydrocephalus in Uganda each year and a likely equivalent number of deaths in neonates. So he goes on to describe what Michele has just told you. He writes, for many reasons, this opens up a range of clinical problems which we now face, and I'm trying to piece together the necessary teams, both within and outside of Uganda, to better learn how to treat and ideally prevent these devastating infections. The organism is highly neurotropic and destructive of the brain, and disturbingly, we have recently seen reported about half a dozen U.S. infants with very similar Penibacillus thiaminolyticus or dendritiformis infections with similar brain destruction and high mortality as in Uganda. And he writes, I suspect you have encountered these cases in Bududa, where Daniel goes every year to work. We are setting up a PCR lab with our primers in Mbale to serve the post-infectious hydrocephalus patients at the Cure Children's Hospital and the regional referral hospital. We would, of course, be happy to analyze your specimens from the Bududa clinic if such testing might be indicated. So after we received this, then Michele decided to do this paper, not knowing that Stephen had written to us. So that's pretty cool.
Very cool. And, I would encourage them to look in their water. I would begin to test the water supplies because this microbe could be hiding out. As Michele knows better than most, Legionella hides out. And Panibacillus could be in that same league of hiding out in our water supplies.
I want to give a shout out to Jennifer Chen, who authored a report of this study in the Yale School of Medicine publication Neurosurgery that I received in an email. So hats off to them for sharing this success and amazing work, collaborative work from across 5 countries. And I'm glad that he's interested in our podcast. And we'll have to let him know we covered it.
Yes. All right. That is TWiM 294. Looks like we're going to hit 300 before the end of the year, folks. That'll be fun. If you, the show notes, by the way, microbe.tv/TWiM. As I said, if you like our work, please support us, microbe.tv/contribute. And questions, comments, TWiM at microbe.tv. Michele Swanson's at the University of Michigan. Thank you, Michele.
My pleasure. Thank you both.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.